Hi everyone. In this lecture series, we are going to briefly discuss the limit test. Limit test are quantitative or semi-quantitative tests that is performed to limit the presence of any impurities present in our pharmaceutical products as well as substances. So, limit tests are considered as semi-quantitative because in limit test we will be using some reagents. Those reagents will react with our impurities to form some products, and those products will be viewed in the form of opalescence or turbidity or some colored complexes. So we will be using a standard solution here in this case, and the standard solution will have some known amount of impurity, and that known amount of impurity will also have chance to produce turbidity, opalescence or in color intensity. And with the help of that standard solutions, we are going to compare our test solution. So we can easily find out whether the impurities present in our sample is within the pharmacopoeial limit or not. So as in the form of this observation, we can be able to identify whether the impurities are present within the limit or not. We are not going to quantitatively measure how much amount of impurity is present, but we are just comparing it with the standard solution, and we are saying whether it fails the test or passes the test, whether it is below the known standard or above the standard. So as we are comparing it like this, it is considered as a semi-quantitative test. But limit test is also considered as a quantitative test because in some cases we will be measuring the impurity with the help of some analytical instruments like nephrometer or turbidimeter or colorimetry. So for measuring the color intensity, we will be using colorimetry or UV visible spectrophotometry. And in some cases, for measuring the opalescence or turbidity, we will be using nephroturbidimeter. So in this, we are going to quantify the impurities. We are going to exactly measure how much amount of impurity is present in our pharmaceutical product. So it is considered as a quantitative test also. So when coming to impurities, what are all considered as impurities? So impurity is something that is unwanted compound that is present in our pharmaceutical product. So those impurities are some undesirable components which when consumed in larger amount for a prolonged period of time, it may induce some harmful effect to our body. So that is why they are considered as impurities, and we are doing this limit test to find out how much amount of impurity is present in our product. So these impurities are not only unwanted compounds, but also sometimes they are toxic components also. So the impurities that may be present in our pharmaceutical products that are found causing some ill effects to our body are listed as fluoride, sulfate, iron, and heavy metals such as lead, arsenic, chromium, and mercury. So these impurities, when present in large amount, may cause some ill effects to our body because when we consider about consuming the products that are having impurities, whatever taken inside our body should have to be eliminated after some time. So these impurities, when we are consuming, it has to be eliminated. But when it comes to this kind of impurities and heavy metals, they are difficult to be eliminated. So it's true, we need minerals like fluoride, sulfates, and some other minerals for consuming, but it has to be present only in a trace amount. If it is present in larger quantities, they are not suitable for consumption. So why performing limit test are considered as advantageous is because by identifying the quantity of impurity present in our products, we can maintain the quality of our product. We can maintain the consistency in quality because we know the impurities are having chances to react with our active pharmaceutical ingredient or excipients. So it may give rise to some undesirable components or degradation products when we store them for a prolonged period of time. So that is why we need to know how much amount of impurity is exactly present and how much the product can withstand its shelf life or its quality until the expiry date. So first and foremost thing is we can able to maintain the quality of our product, and we can maintain the consistency over batches, and then we can also prevent the presence of impurities in our consumable products that will be present more than the desirable amount, so we can consume them as per the recommended allowances. So in the further videos, we will be seeing about the various limit tests and how to perform them and what are the advantages of it. Thank you.